everyone, welcome back to The Explainer. So, we did it. We finally made it out of the energy investment phase of glycolysis. I mean, we've spent our 2 ATPs, we've literally split our sugar molecule right down the middle, and now it's time to actually get a return on that molecular investment. Today, we're looking at reaction 6. This is a totally pivotal moment because it's the very first time we harvest energy, not as ATP just yet, but as high-energy electrons. We're also going to see how a deceptively simple element like arsenic can completely sabotage this beautiful biological engine. So let's jump right into the chemistry. Here's our quick roadmap for today. First, the energy generation phase, then the mechanism of reaction 6, followed by the NAD+ bottleneck. After that, we'll hit the arsenate poisoning sabotage and wrap it all up with memory aids and an exam review. Let's go.
All right, Section 1, the energy generation phase. Think of our cellular pathway as a biological factory assembly line. We're basically entering the phase where the factory finally starts paying us back. And you got to remember, from this point forward in glycolysis, everything, and I mean everything, is happening twice for every single molecule of glucose we started with. Why? Well, because we split that 6-carbon sugar in half earlier, right? So we currently have 2 3-carbon molecules of glyceraldehyde 3-phosphate, or G3P, ready to roll down the line. Which brings us to reaction 6. This is the conversion of our G3P into 1,3-biphosphoglycerate, or 1,3-BPG. And what's really interesting here is that this reaction is totally unique compared to everything we've seen so far. It's the very first oxidation reduction, or redox reaction, in all of glycolysis. This is the exact moment the cell finally starts extracting raw energy, specifically in the form of high-energy electrons.
Moving on to Section 2, mechanism of reaction 6.
Okay, so this whole transformation is catalyzed by an enzyme called glyceraldehyde 3-phosphate dehydrogenase. Now, starting with our G3P, the enzyme needs an oxidizing agent to actually pull this off. It basically needs something to act as a catcher's mitt for the electrons it's stripping away. And that molecular catcher is NAD+. NAD+ steps right in, catches those electrons, and boom, it gets reduced to NADH.
But let's look a bit closer at what the enzyme is actually doing to the molecule itself, because it's doing 2 things at the exact same time. First, it oxidizes the aldehyde group on carbon 1 of the G3P molecule straight into a carboxyl group. Then second, it immediately grabs a free-floating inorganic phosphate. And get this, that's not a phosphate from ATP, just a free unattached phosphate. And it sticks it right onto that brand new carboxyl group.
Now, you might be wondering, why does it happen exactly like this? Honestly, it's an absolute masterclass in energy engineering. See, the oxidation of that aldehyde group releases a massive amount of free energy. If the cell wasn't incredibly careful, all that energy would just vanish as heat. Gone. Instead, the cell smartly couples that energy release to the attachment of the phosphate. So the resulting bond on carbon 1 of our new molecule, 1,3-BPG, becomes a super high energy bond. It basically traps and stores the energy from the oxidation, setting it up perfectly to synthesize ATP in the very next reaction.
All right, Section 3, the NAD+ bottleneck.
But, of course, there's a catch. Your cells actually have a very limited, finite supply of NAD+ just sitting around in the cytosol. Think of NAD+ molecules as a fleet of empty shuttle buses for electrons. If you just keep running glycolysis, continually stripping electrons and loading them onto the buses, well, eventually you're going to turn all your NAD+ into NADH. Every single bus will be packed full. And if you run out of empty NAD+ buses, reaction 6 stops dead in its tracks. The whole energy production line literally grinds to a halt. So to prevent a total catastrophic shutdown, the cell has to rapidly recycle that full NADH back into empty NAD+. It needs to empty the buses. The body has 2 main ways to pull this off. Aerobically, so when we have oxygen, it can send the NADH straight to the electron transport chain in the mitochondria to drop off the electrons. Anaerobically, though, like when oxygen is scarce during an intense workout, the cell uses the NADH to reduce pyruvate into lactate using an enzyme called lactate dehydrogenase. Both methods empty the bus so glycolysis can keep right on running.
Which brings us to Section 4, arsenate poisoning sabotage.
It's fascinating and honestly a little bit terrifying to think about. How does a completely simple elemental imposter manage to bypass our perfectly tuned biological safeguards to just shut down our energy harvest?
Well, the answer is molecular mimicry. Pentavalent arsenic, which we call arsenate,
looks almost exactly like a normal, harmless, inorganic phosphate molecule. Because they look so incredibly similar, our hardworking enzyme, glyceraldehyde 3-phosphate dehydrogenase, simply can't tell them apart. It grabs the toxic arsenate molecule instead of a phosphate and just sticks it right onto the G3P. But here is the fatal flaw. That bond between arsenate and carbon-1, it is highly unstable. It spontaneously hydrolyzes, meaning it just breaks apart with water to form 3-phosphoglycerate. Think about what just happened there. We completely bypassed the synthesis of our actual target molecule, 1,3-BPG.
And this perfectly illustrates the real danger. Since we never made 1,3-BPG, we have 0 high-energy phosphate bonds to transfer over to make ATP in the next step. I mean, the enzyme did the work, the cell ran the reaction, but it gets absolutely no energy payoff. The cell is utterly deprived of the energy it was supposed to harvest, and that right there is exactly why arsenate poisoning is lethal. It doesn't stop glycolysis from running. Instead, it maliciously shorts out the energy harvest, basically leaving the cell to starve while the engine is still spinning.
Okay, let's wrap things up with Section 5, Memory Aids and Exam Review.
If you want a totally foolproof way to remember the clinical correlation for arsenate poisoning, just think of it like a counterfeit coin at a vending machine. The coin, the arsenate, is shaped perfectly. It fits right into the enzyme machine. The machine accepts it, processes it, but because the coin is fundamentally fake, the machine doesn't actually drop the prize at the bottom. You walk away with absolutely no ATP.
Let's hit a few rapid-fire review questions. I definitely recommend pausing the explainer right here if you're prepping for exams. Ask yourself, can you clearly explain why reaction 6 is considered a redox reaction? Remember that electron transfer to NAD+? What happens if a cell fails to regenerate NAD+? Well, the whole factory shuts down. How is the energy of the aldehyde oxidation trapped? Through that high-energy phosphate bond coupling. And finally, how exactly does arsenate alter the net ATP yield? By bypassing 1,3-BPG, it drops our net ATP production significantly.
It really is an incredible system, but as we've seen, it's kind of a fragile one too. Arsenate uses molecular mimicry to slip right past our defenses and literally rob us blind. Which leaves me with a final thought for you. If our biological factory is so intricately, brilliantly engineered, what other simple, everyday elemental imposters might be secretly exploiting our pathways right now? Definitely something to think about. Thanks so much for joining me for this explainer. Keep reviewing those notes, and I'll catch you in the next one.
Hello, everyone. In our last lesson, we saw how the cell harvests high-energy electrons and traps energy in a molecule called 1,3-BPG. But up until now, we've actually been in a financial hole. We've spent 2 ATPs and haven't gotten any back. Today, that changes. We are looking at reaction 7, the moment we finally break even, and a very special side path that your red blood cells take to help you breathe. Let's get into the first real energy harvest.
Here is the roadmap for our explainer today. First, finally breaking even. Second, substrate-level phosphorylation. Third, the red blood cell side mission. Fourth, trading energy for function. And fifth, exam prep and memory aids. Let's jump in.
Section 1. Finally breaking even. Up until this exact moment in the glycolysis pathway, we've been spending capital to make money. We invested 2 ATPs early on to prime the glucose molecule, and so far, we've seen 0 return on that biochemical investment. We are sitting firmly at a net of -2 ATP.
In reaction 7, we take that high-energy 1,3-bisphosphoglycerate we just made and convert it into 3-phosphoglycerate. This reaction is catalyzed by the enzyme phosphoglycerate kinase. Now, don't let the name kinase confuse you. Usually, kinases add phosphates to things, but in this specific case, the enzyme takes a phosphate off of the 1,3-BPG and hands it directly to ADP to create ATP. Imagine your 3-carbon molecule with a phosphate on carbon 1 and carbon 3. The enzyme reaches in, snaps the phosphate off carbon 1, and sticks it onto an ADP molecule. What's left is a 3-carbon molecule with only 1 phosphate remaining on carbon 3. Because this reaction happens twice for every glucose molecule, we generate 2 ATPs here.
This is a huge milestone. These 2 newly minted ATPs perfectly repay the 2 ATPs we spent way back in the energy investment phase. As of this moment, our net ATP gain for the whole pathway is exactly 0. We've officially broken even.
Section 2. Substrate-level phosphorylation.
This reaction is a textbook example of substrate-level phosphorylation. It's really important to understand how this differs from what happens later on.
In most of your metabolism, ATP is made in the mitochondria using a complex wind turbine called the electron transport chain. That's oxidative phosphorylation. But here, we aren't using a turbine. We are taking a high-energy phosphate directly from a substrate, R13-BPG, and giving it to ADP. It's a direct one-to-one molecular handoff.
Here is a fascinating detail for your exams. Most kinases in glycolysis are irreversible one-way streets. However, phosphoglycerate kinase is physiologically reversible. This means if the cell has a massive surplus of ATP and 3-phosphoglycerate, the reaction could theoretically run backward. Of course, in the context of burning sugar, it almost always moves forward toward energy production, but chemically, it definitely has that reversible flexibility.
Section 3. The red blood cell side mission.
Now, let's talk about a special side mission that happens only in red blood cells. This is called the Rappaport-Lubbering glycolytic shunt. Your red blood cells have a unique priority. While other cells just want ATP, red blood cells are the body's delivery trucks for oxygen. To do their job, they sometimes divert 1,3-BPG away from the main highway of glycolysis.
So, how does this actually work? Instead of going through reaction 7 to make ATP, an enzyme called bis-phosphoglycerate mutase takes 1,3-BPG and moves the phosphate from carbon 1 to carbon 2. This creates a molecule called 2,3-bisphosphoglycerate, or 2,3-BPG. Why do this? Because 2,3-BPG is a master regulator of your blood. It binds to hemoglobin and decreases its affinity for oxygen. In plain English, 2,3-BPG tells your blood to let go of the oxygen so it can enter your tissues. Without this molecule, your hemoglobin would be too greedy, holding on to oxygen so tightly that your muscles and brain would suffocate.
Notice exactly how the red blood cell physically branches off and bypasses the standard phosphoglycerate kinase step. The 1,3-BPG goes off to become 2,3-BPG via the mutase, does its vital job helping hemoglobin drop off oxygen, and then eventually a phosphatase enzyme removes a phosphate to turn it back into 3-phosphoglycerate. At that point, it rejoins the main glycolytic path. Section 4. Trading energy for function.
There is a real cost to this detour. When the red blood cell takes this shunt to make 2,3-BPG, it bypasses the phosphoglycerate kinase reaction entirely. This means no ATP is produced during this step. Eventually, the 2,3-BPG is converted back into 3-phosphoglycerate and it rejoins the main glycolytic path. But notice the incredible result here. The red blood cell is traded energy for function. It sacrifices the ATP it could have made so that it can ensure your tissues get enough oxygen. This is a beautiful example of biological prioritization.
Section 5. Exam prep and memory aids.
Let's lock this in before we finish. Think of reaction 7 as your payday. You've been working hard and spending money, and now you finally get your cash, your ATP back. Now think of the red blood cell shunt as a charity donation. The red blood cell is the donor who says, I could keep this money for myself, but I'm going to spend it on a gift, 2,3-BPG, that helps the rest of the body breathe better.
All right, let's test your understanding with some active recall. Explain the breaking even concept at the end of reaction 7 in terms of ATP accounting. Think about our initial investment versus what we just harvested.
Exactly. The 2 ATPs produced here perfectly repay the 2 ATPs spent during the energy investment phase, making our net ATPs 0. We climbed right out of that initial financial hole.
Next question. Why is the 2,3-BPG shunt described as a bypass of the energy-generating phase? What exactly is being skipped? It skips the phosphoglycerate kinase reaction entirely, meaning 0 ATP is produced during this specific step. It's an absolutely massive physiological sacrifice.
Let's do a rapid-fire recall question. Name the specific enzyme inside red blood cells that converts 1,3-BPG into 2,3-BPG.
The enzyme you need to memorize is bis-phosphoglycerate mutase. Remember, a mutase just relocates a functional group within the exact same molecule.
We'll end on this final provocative thought. What would be the devastating physiologic consequence if a red blood cell was unable to produce 2,3-BPG? Would your tissues actually suffocate? Think about hemoglobin's natural greediness for oxygen and how this one tiny molecule created by a glycolytic detour keeps every single cell in your body breathing. It's a perfect example of the sheer genius of cellular metabolism. Thanks for joining me for this explainer, and keep exploring the amazing world inside your cells.
Welcome back to The Explainer. You guys, we are finally in the home stretch of the glycolysis factory line. In our last lesson, if you remember, we successfully broke even on our initial ATP investment. But now it's time to move past that break-even point and race straight toward a net profit. Today, we're covering the final 3 reactions of the pathway, 8, 9, and 10. We'll see exactly how the cell meticulously rearranges its molecules to create a super high-energy compound, and we're even going to discover how the fluoride in your toothpaste actually works at a biochemical level. Let's finish this race to pyruvate. Okay, let's dive right into this molecular assembly line and see exactly how the cell rearranges its substrates to squeeze out that final high-energy payoff. We've invested energy, we've split our sugar, and we've recouped our costs. So now, every single molecule of ATP we generate, that is pure, unadulterated metabolic profit for the cell. And here is our roadmap for today, covering reactions 8, 9, and 10. We're starting right at the top with 3-phosphoglycerate, which, you might recall, was the product of our very first payday back in reaction 7. By the time we reach the bottom of this sequence, we're going to have transformed it into our ultimate product, pyruvate.
So let's kick things off with reaction 8, molecular housekeeping.
Now, this step isn't going to yield energy just yet. It's essentially a bit of mandatory molecular housekeeping on the factory floor, just getting things prepped for the heavy lifting to come. An enzyme called phosphoglycerate mutase steps in. Imagine your 3-carbon chain. Currently, the phosphate, which you can kind of think of as a bulky molecular backpack, is attached to the bottom carbon, carbon 3. The mutase enzyme just picks up that backpack and shifts it up to the middle carbon, carbon 2. This gives us 2-phosphoglycerate. Simple, right? It's a freely reversible step, meaning it's not a major control point. It's literally just moving things around to prime the molecule for what's next. Moving right along to reaction 9, building the pressure. Here's where things get really interesting. We take that perfectly primed molecule and suddenly crank up the tension. An enzyme called enolase swoops in and performs a dehydration reaction, literally pulling a molecule of water right out of 2-phosphoglycerate. By pulling out that water, enolase acts like a vice, redistributing the internal energy of the substrate. It tightly coils the molecule into a spring called phosphoenol pyruvate, or PEP. And PEP is a massive deal because it contains a remarkably high-energy enol phosphate bond. Even though this reaction is freely reversible, PEP is one of the highest energy molecules in all of biology. Seriously, it's higher energy than ATP itself. It's like a biological pressure cooker, squeezed as tight as it can possibly go, just waiting to release that energy. Which brings us to a quick clinical spotlight, throwing a wrench in the gears. Before we release that molecular spring, let's look at a really cool clinical correlation. What happens when an everyday chemical jams up this machinery? I'm talking about fluoride. Fluoride actually inhibits the enzyme enolase, effectively halting the glycolytic factory line dead in its tracks right at reaction 9. And that is exactly why it's in your toothpaste. When you brush, it stops the bacteria in your mouth from finishing glycolysis, which prevents them from producing the lactic acid that eats away at your tooth enamel. Plus, in the hospital lab, we use fluoride and oxalate in blood collection tubes. This literally freezes the blood cells' metabolism so they don't consume the blood sugar while the tube sits there waiting for the lab. That ensures the glucose number the doctor reads is exactly what was in the patient's veins. Pretty neat, right?
Okay, back to the pathway. Reaction 10, the second payday.
With the pressure fully built up in PEP and our machinery humming along, it is time for the big payoff. Now, it's really important to contrast the nature of these final steps. Reactions 8 and 9, they were casual, freely reversible steps, mostly focused on molecular positioning and building pressure. But reaction 10 is strictly irreversible. The conversion of PEP to pyruvate is catalyzed by pyruvate kinase, or PK, and this acts as the third heavily guarded, completely irreversible gateway of the entire glycolysis pathway. So, the crucial point here is how this actually affects our overall energy ledger. Pyruvate kinase acts as the ultimate release valve for our molecular pressure cooker. It snaps that super high-energy phosphate off of PEP and hands it directly to ADP to create ATP, which is a process called substrate-level phosphorylation. And because our original glucose was split into 2 3-carbon molecules way back when, this reaction happens twice per glucose. Let's check the ledger. We spent 2 ATP initially, we gained 2 back at reaction 7, and now we gain 2 more right here at reaction 10. Boom! We have finally achieved a net profit of 2 ATPs for the entire pathway. And pyruvate is our normal end product here under aerobic conditions. Now let's talk about regulating the factory line. Because pyruvate kinase acts as this irreversible release valve, dumping all that built-up energy to finalize the pathway, the cell absolutely has to tightly control when it operates. It handles this through a brilliant mechanism called feed-forward activation. Do you remember PFK1 from lesson 2? Well, when PFK1 is highly active early on, it produces a massive amount of fructose 1,6-bisphosphate. This molecule actually travels all the way downstream to the end of the line and activates pyruvate kinase. It's exactly like the front of the factory calling the back of the line to shout, "Hey, a huge shipment is coming, get ready to work." This guarantees the whole system stays perfectly synchronized. All right, time for some exam review and memory aids. We've covered the mechanics, the clinical ties, and the regulation. Let's lock this knowledge in by tackling some high-yield exam questions you are practically guaranteed to see. First up, why is the dehydration step by enolase so absolutely necessary for the subsequent production of ATP? Think back to our pressure cooker metaphor for a second. Pulling that water out is exactly what coils the molecular spring. It redistributes the internal energy to create PEP, forming a wildly high-energy enol-phosphate bond. That intense molecular tension is what gives PEP the immense transfer potential needed to forcefully attach a phosphate onto ADP in the very next step. Okay, let's do a quick rapid-fire recall question regarding our earlier molecular housekeeping step. Which enzyme catalyzes the shift of the phosphate group from carbon 3 to carbon 2? The answer is phosphoglycerate mutase. Just remember the name itself gives it away. A mutase simply mutates the position of a functional group. It's like moving a backpack from the bottom to the middle without changing the overall chemical formula of the molecule. Next question. How does fluoride's effect on enolase translate to its use in clinical blood glucose testing? This directly connects our biochemistry to the patient's bedside and those lab results. Well, by inhibiting enolase, fluoride effectively stops glycolysis altogether. Using a fluoride and oxalate tube literally freezes the metabolic factory line in the drawn blood. It prevents the red blood cells from consuming the glucose, ensuring the number on the patient's chart is perfectly accurate. To make these final steps completely unforgettable for test day, think of PEP as a pressure cooker. Reaction 9 builds up the pressure, and Reaction 10 is the release valve that captures all that escaping steam as ATP. And regarding clinical inhibitors, just remember this simple, catchy rhyme. Fluoride stops the enolase flow so the cavities don't grow. If you can remember that, you're going to nail any question on Reaction 9. And just like that, it brings us to the end of the line. We hold pyruvate in our hands, the ultimate end product under normal aerobic conditions, alongside our sweet net profit of 2 ATP. But our biochemical journey is far from over. Where does this pyruvate actually go next? What triggers the cell to push it into the mitochondria for massive energy yields versus fermenting it into lactic acid? And how does that single metabolic decision dictate human survival during intense exercise or oxygen deprivation? Well, we'll unravel that mystery in our next explainer. Keep learning, and I'll see you then.